business of this evening. The Society's highest award each year is the National Lecture. And this year's National Lecture, I'm very pleased to announce, is Karolin Luger from Colorado State for her work on the nucleosome structure. She was the first author on the important paper for the first crystal structure of the nucleosome. And at Colorado State, her lab had done many more structures and a lot of other work connecting the changes and both structural changes and functional changes in the nucleosome, and has also spread that work to other techniques besides crystallography. And I'm sure you will get a really interesting lecture. So I want to turn it over to Karolin Luger after presenting her with this plaque. So I guess we can leave yours up here.
Thank you.
Great. And you can grab it on the way down.
All right. I'm the only thing standing between you and the dancing. And so let's get this show on the road. So thank you, Dr. Richardson, Jane, and the Biophysical Society for this incredible honor and for this wonderful opportunity to address so many biophysicists at the same time. This is a pretty amazing sight here from where I'm standing. Is also pretty humbling because I'm well aware of the cumulative knowledge and creativity and wonderful science that's sitting right here before me. And I realize to a certain extent it's probably a little arbitrary that I am standing here, but I definitely would not be standing here, sorry, this is my title, if it weren't for my wonderful lab members, past and present, starting from this rather forlorn-looking group of my 4 first graduate students stranded on the side of the road. I'm sure some first and second year students can relate to that picture. To a rather happily growing bunch in 2003, and then this gang here is clearly a little out of hand and having way too much fun. But of course, all this growth, all of these individuals are just marvelous, and they contributed in their own way to the success of the lab and to each other's success. And I really take this award on their behalf. And all this growth, of course, wouldn't be possible without wonderful funding agencies who, especially in the early stages of my career, placed a lot of faith in what I was proposing to do. So thanks to them as well. But as scientists, we're all products of our mentors. And I was really blessed with wonderful mentors, starting from my early first stumbling steps in the lab at the University of Innsbruck, where people were very patient with me. Bernd Auer mentored my first DNA preps, very exciting times. My PhD work with Professor Kirschner at the Biocenter in Basel was indeed the most fun I think I've ever had in science, because he was very creative and he let us follow our own pretty crazy ideas with sometimes lucky outcome and sometimes not so much. And of course, Tim Richmond and David Sargent at the ETH Zurich, who taught me everything I needed to know about crystallography and about chromatin structure. But especially in the early days of my career, I was really fortunate to have good friends and mentors who would give me open advice, would give me critical advice, and I'm very, very grateful to them. And so this, I guess, is my passionate pitch for mentorship. I wouldn't be here without these wonderful people. And all those of you, graduate students who grudgingly take on this bumbling undergrad who can't do anything right, just remember that you hold these people's careers in your hands, and maybe sometime one of your undergrads will stand up here and show your picture here on the board. So keep on mentoring, because it's a very, very important part of our job. All right, so let's get to the business at hand. What I thought I would do, since I have so many of you before me, there's a number of things I want to say about nucleosomes and the history of the nucleosome structure, because I get asked this a lot. Now, when I planned this lecture, I did not know that we would be hit in the face with the nucleosome structure pretty much every turn in in the hallway, so I apologize for the nucleosome overload, but just bear with me here. I will then talk about more recent work on epigenetic variations of nucleosome structure, and this really drove home the fact that we needed to use hybrid approaches to get at these difficult problems. And then finally, if I have the time, I'll talk about the thermodynamics of nucleosome assembly and disassembly. So why are we interested in nucleosomes? All of us who are sitting here are very complex organisms, and complex organisms call for complicated genomes. So the genome of each and every one of your cells, if pieced one end to end, would be about 2 meters in length. The nucleus is very much smaller, about 5 times 10 to the minus 6 meters in diameter. And so to put this into an analogy, you would have to take a 10-mile thread, very thin thread, and put that into a golf ball. Now this is actually doable, I think, but this whole problem is aggravated by the fact that DNA encodes information in linear form, pretty much like these thankfully long forgotten music cassette tapes that I certainly remember, but many of you probably don't. We have problems with these tapes, as I also do remember. And this here on the right is our unfortunate systems manager who's trying to I don't actually know what he's trying to do, but it is a computer backup tape. So the requirements for genome packaging poses, there are many challenges to this, as you can see. We need to package the information in a very confined space. That's the first order. We need to protect it from physical damages and tangles. We need to regulate access to the stored information, and this is the hallmark of regulated gene transcription, which is extremely important to all life in our cells. And we need to accurately and rapidly replicate the genome, again, without getting tangles and knots. Nature's approach to this is actually deceptively simple. So this is linear B-form DNA that forms a complex with an equal mass of protein. And these assemblies are collectively called chromatin. So for every gram of DNA, there's a gram of protein in our cells. The majority of these proteins are the histone proteins called H2A, H2B, H3, and H4. This is very old terminology. These histones form an octamer, a tiny little protein spool around which we wrap small segments of the DNA. And these are the nucleosomes. And about 100,000 of nucleosomes are then arranged in a beads on a string-like fashion and then further compacted into manners that we don't understand quite yet. There's additional non-histone proteins that are very important to further regulate chromatin compaction. Now, if we stay with this somewhat nucleosome-centric view of the genome for a little while longer, you will realize that these are very important structures. They determine the spatial and temporal access to the genome because they organize the entire DNA. So every DNA, every base pair of DNA in our bodies is organized in these structures. And as such, they inhibit access to the DNA and they regulate transcription, replication, and DNA repair, basically all processes that require access to the DNA. They maintain genomic stability, so they protect the genome against breaks and regulate DNA repair, and they're also the targets of epigenetic control, and we'll talk a little bit about that later. This view of chromatin and the nucleosome has not always been accepted, and it was based on early electron micrographs by Chris Woodcock and some others showing these tiny little beaded structures here. And this quote is from a paper review for one of Chris Woodcock's paper saying that a eukaryotic chromosome made out of self-assembling 70 angstrom units, which could perhaps be made to crystallize, would necessitate writing our basic textbooks. I have never read such naive paper purporting to be of such fundamental significance. And then this review went on saying, definitely it should not be published anywhere. So you're laughing now, but unfortunately, the publication of this paper indeed was delayed by 1 year through this review. But luckily, it did not stop research on these important structures. And in 1984, Tim Richmond and his colleagues in Aaron Klug's lab determined the 7-angstrom structure of the nucleosome. And shown here is a nature cover with the electron density maps, the 7-angstrom electron density maps, a section of these structures. Now, as you probably all know, 7 angstroms is not really enough to distinguish side chains or base pairs. But the map was actually of astonishing quality, and it was quite easy to distinguish protein from DNA because of the higher scattering mass. But the interpretation of this map and the assignment of the protein chains was greatly aided by some very, very thorough biochemistry from Mirza Bekov and colleagues using histone DNA cross-linking in a very meticulous fashion. So we have a 7-angstrom electron density, and this obviously needs to be improved. How do we do this? The first order is sample homogeneity. That's always what crystallographers do. When something produces bad crystals, we try to improve the purity of our compounds. The second problem we have is the phasing of large macromolecular complexes, which is still not a trivial problem. And the third problem at that time was instrumentation. And this was at a time when third-generation synchrotron instruments basically just came online. Sample homogeneity was a very obvious one to address. The way these original crystals were obtained is by chopping up chromatin from calf thymus or erythrocytes, and necessarily these histones from natural sources were very heterogeneous. They had different post-translational modifications, they had proteolyzed chains. It was just quite messy when they were analyzed on gels. And furthermore, because nucleosomes organized the entire genome, the DNA sequence and also the DNA length was quite non-uniform, especially the DNA sequence. So this would immediately limit resolution. So it was very clear that we need nucleosomes with defined sequence and with recombinant histone proteins, so-called recombinant nucleosomes. So how do we do this? Tim Richmond in 1988 devised a really nice system with the help of Song Tan, where they duplicated up 147 base pairs of DNA and into plasmids. And so what we had to do was basically prepare 12 liters or 24 liters of bacterial culture, then do mini preps. There was no kit then, so there's a lot of phenol that's involved. And through a series of purification steps, obtain milligram amounts of defined sequence 147 base pair DNA. Or we could also prepare arrays, N-mers of the same DNA. The histones had to be cloned into separate expression vectors and expressed separately in E. coli cells, then purified from inclusion bodies, and then refolded into histone octamers, which would then be used to assemble nucleosomes. Now, this system has actually initially was very tedious, but it proved to be extremely versatile because it allows us to make mutations in any histone protein we want, to do substitutions, to do tail deletions, to introduce histone variants, and it was essential for the phasing of this structure, as you will see. And I think the vectors that I made originally are still, are probably in use in every chromatin lab that I know of. So this is our contribution to chromatin research. All right, so we take these components, we put them together by salt gradient reconstitution to make either mononucleosomes or nucleosomal arrays. The mononucleosomes, in theory, would produce these wonderful crystals and everybody would be happy after. And the nucleosomal arrays could be used to do atomic force microscopy and analytical ultracentrifugation. And we've done a lot of research on this, but for now I will restrict myself to the mononucleosomes. All right, so these crystals, that was the expectation. This was the reality. So, in fact, these recombinant nucleosomes were worse than the native ones that we had used before. They crystallized as these thin, horrible window pane crystals, no real apparent thickness to them. The diffraction actually deteriorated. On a good day, maybe we got 4.5 angstroms. On a bad day, we got 10 or 15. And what was the most annoying, they were very slow and very unpredictable in crystallization, and we couldn't really quite get a handle on why they would crystallize and why some wouldn't crystallize. So then I think I did what I still consider the most important experiment of my career. I took these miserable little things and washed them and dissolved them and analyzed them on a native gel. And this is what you see. So after very long scientific analysis, you will realize that these guys here to the very right are fuzzy. That's the input material. That didn't bother me that much because that's what nucleosomes always were. Everybody in the lab said on native gels, nucleosomes are fuzzy. Okay, they're fuzzy. But my dissolved crystals were not fuzzy. They were a nice sharp band that ran a little bit lower. So I tinkered with the gel conditions for a little bit. And indeed, what you can see after very thorough scientific investigation is that the fuzzy nucleosomes are in fact 3 bands on the native gel. So these are the same gel systems, just slightly improved. Whereas the crystals that are dissolved, denoted with an X here, are in the lowest band and they form a single band, invariably. What you can also see is that our mother liquor is empty. No nucleosomes in the mother liquor. So what this told us is that the crystal selectively takes the lowest band and incorporates that, and maybe the upper 2 bands either get destroyed or our 2 middle bands get transformed into the lower band through the process of crystallization. So this is something to be followed up on. Through some biochemistry that I don't want to bore you with, we actually discovered that these 3 bands, or in some cases 2 bands, represent alternative positions of the DNA on the histone octamer. So in these upper bands, pieces of DNA were sticking out on one side and they were missing at the other side. And that was enough to cause differences in their migration. And the lowest band were these central nucleosomes in the central position here. So it seemed that the crystal really liked to select for the central position. So this was selected for by the crystallization process. And luckily, through some more or less, how shall I say this, fortuitous experiment, I discovered that you could transform these upper bands into the lower band by a simple heat shifting assay in a non-dissociative manner. So I got these nucleosomes to all shift down to the central position. This step alone, together with the identification of a palindromic DNA sequence that had very strong positioning propensities, were the key towards high diffraction. And this last point was painstaking work by Song Tan and by Armin Maeder, who analyzed a lot of sequences and heat shifted them and analyzed them for their diffraction quality. And lo and behold, finally we got our wonderful crystals, but as crystallographers, we knew that this is not necessarily the key to high diffraction because often the most beautiful crystals don't diffract very well at all. Luckily, in this case, these guys diffracted pretty well, but we had to do some fancy dehydration and cryocooling protocols to get them to do their thing, and I'd be happy to talk about these technicalities to anybody who's interested. So my point is, if your crystals don't diffract very well, don't throw them away, but tinker with them a little more just to make them, to maybe get the most out of them. All right, so Kurt Davies subsequently improved these crystals further to this stunning diffraction pattern of about 1.9 angstrom. But to me, and my message to you guys is the most important crystallography experiment is to dissolve your crystals, to analyze them by every imaginable method, and to compare them with the input. Because these crystals, as miserable as they may be, they are talking to you, and you have to listen to them. And may that be proteolysis, may that be some conformational changes. If something crystallizes slowly, there's usually a reason for it. And if it works for you, please let me know. Okay, so we've gotten over the first hurdle. This was at this stage me trying to push this nucleosome up a hill. We've licked the substrate homogeneity, we've got diffracting crystals. Now we needed to phase these crystals. And this was back in a time where SAD phasing or MAD phasing was not really standard, and it wouldn't really work with these large structures at that time. And so we had to go in and painstakingly engineer about 20 cysteine substitutions throughout all the histone sequences, derivatize those with heavy atom clusters, and then do multiple isomorphous replacement. And those of you in the room who've done this know that this is a rather tedious process and quite frustrating, but finally we ended up with about 5 derivatives that gave us phases that were passable, not great, but passable. And so after about 1 year, burying myself in the dark room, literally, we finally managed to solve the structure to 2.8 angstrom resolution. And at this resolution, as you know, individual atoms are visible. I shouldn't say that. There's too many physical chemists here. We can see side chains and we can see base pairs. So we actually can now see how this structure is held together and what the molecular details of the contacts are. All right, so this is the nucleosome crew. Tim Richmond, of course, here with a beautiful model, 3D model of the nucleosome, who kept the faith in this whole project throughout all the year, and then especially Song Tan and Armin Maeder and Thomas Rechsteiner, who were my partners in this long and arduous undertakings. And a lot of the credit also goes to the synchrotron, then barely out of construction in Grenoble, and Christian Riekels and Stephen Cusick's beamlines, and these guys were very, very helpful. All right, so since I have you all sitting here, Nucleosome 101. What should you guys know about these structures that hang everywhere? This is actually not a very large structure by today's standards anyway. It's about 110 times 55 angstroms. Molecular weight is about 210 kilodalton. The structure consists 50% of protein, 50% of DNA, and the DNA is wrapped around this outside of a protein cylinder in these 1.65 superhelical turn. Now, it's a rather convoluted structure, so the best way to understand this is by following its assembly pathway. So we start out with the H3H4 tetramer in blue and green here. If we look at it from above, or if we lie on the floor, these are the views on either side here. And this structure binds about 60 base pairs of DNA. And it follows the outer perimeter of this twisted horseshoe-like arrangement. It's only to this structure that the H2A, H2B dimer binds. Okay, so we get one binding here, and you can see here especially well how this generates additional DNA binding sites for the DNA to continue its superhelical path. So we do this, about 40 additional base pairs here and here, and the same happens in the back. Nucleosomes are symmetric structures, so we have the same procedure happening on the back here. And voila, we have our nucleosome in its full glory in its 3 orthogonal views. So remember that nucleosomes, I want you to remember that nucleosomes are, the histone octamer, the protein part, is not a single entity, but it is in itself a modular assembly. That's important to remember. Nucleosomes are symmetric structures. So basically, if we cut it where I drew this little arrow up here, if we take a scissor and cut the DNA and then slide the 2 halves apart, you will get the 2 halves of the nucleosome slid apart. And this is pretty much the analogy as an Oreo cookie, and you would slide it apart. And then on the left side, you have the filling side that's normally buried at the inside, and this would, on the right-hand side, this would be the outside. All right, so what is much more obvious in this view is that the DNA is actually still B-form DNA, but is very distorted. In addition, protein DNA contacts happen over the entire length of the DNA, every time the minor groove touches the histone octamer. So every one of these little circles I drew here is its own little universe of protein DNA interactions. And I've just shown you a little example here. This is one particular region. And so there's a variety of main chain phosphate interactions. There's water-mediated contacts. There's even some hydrophobic contacts. So I want you to remember when you think of nucleosomes and proteins that want to come in and take the DNA off, it's not a simple task of just breaking open a couple of hydrogen bonds or salt bridges. This is a pretty major undertaking with a lot of buttons, Velcro buttons along the line. So just to summarize what I want you to take away from histone DNA interactions in the nucleosome, they happen exclusively at the minor groove. They happen over the entire length of the DNA. There is no base-specific contacts, and that is important because they organize the entire genome, so they shouldn't be terribly discriminatory towards sequence. They're dominated by main chain phosphate interactions, opposed to popular belief back then that it would be mainly basic side chains that interact with the phosphates. And the DNA is highly distorted, but still mostly in the B-form DNA. Although after listening to Hashim's talk, I want to go back and see whether we can find some Hookstein bases in there. I'm pretty sure some of the DNA looks pretty sick, so I'm pretty sure we can find some if we look. And the histone octamer itself is a modular assembly. All right, so crystallography does not reveal everything. Almost every structure in the PDB is not the entire protein that you put in. And that's a well-known fact. In the case of the nucleosome, it's about 20% of the amino acids that are not visible. And these are the flexible histone tails. So in this view, I've simply stripped away the DNA from the histone octamer. And I've extended in these dashed lines the rough length of the tails that we do not see. These little lollipops here are epigenetic modifications, and there's actually many more than this, and so the histone tails are the sites of epigenetic modification. What does that mean? Let's talk about epigenetic of the nucleosome structure. Now, all of us, every cell in our body has the exact same genome, okay? So from a fertilized egg cell, we produce all these amazing cell types here that look very, very different, and all of these cell types invoke different gene expression patterns, and ultimately it results in an amazing human being like my daughter Maya here at age 2. So the mechanism by which these different gene expression patterns are invoked are through epigenetics. What does epigenetics mean? Epigenetics literally means above genetics. They're chemical changes to the DNA and to the histones. And they're thought to affect nucleosome and chromatin structure. And this was a proposition that we wanted to test. Epigenetics is very much in the mainstream. It started to arrive in the mainstream around 2009, 2010. And Time magazine had this whole article about why DNA isn't your destiny, because the epigenome is affected by lifestyle, nutrition, and environment. And the new science of epigenetic reveals how the choices you make can change your genes and those of your kids. So here is yet another reason for us parents to feel guilty for the horrible things we do to our children. That's all we need. Now, what are these epigenetic regulators of chromatin structure? The most commonly mentioned ones are post-translational modifications, or PTMs. Virtually every modification known to man, known to happen on proteins, happen on the histones. And there's a number of them. And there's an increasing variety of them. More and more sites are being identified. Most of them happen on these flexible histone tails. Additional epigenetic modifications actually are the incorporation of histone variants. I'm just pointing out SEN-H3, which I'll talk about a little later. And this is a histone variant that exists only at the centromere of each chromosome. So we'll talk a little bit about that later. And then there's other histone variants as well, but DNA sequence and DNA methylation, of course, is also very, very important epigenetic modification that is responsible for gene silencing in a very interesting manner. Now, if you need to have any more motivation to follow me through this section here, just realize there recently has been a book published that you can, through the new science of epigenetics, you can reveal the happiness genes in your DNA, okay, unlock the potential hidden in your DNA. So I should say that there's no commercial interest, and I had nothing to do with this book, and I did not read it, but I just thought it would be, and I'm not sure I should read it, but I'm saying that this book probably sells more copy than any textbook on epigenetics. All right, joking aside, let's talk about epigenetics. So the question we posed was, how do epigenetics modifications affect nucleosome structure? So we set about doing crystallography on a number of different nucleosomes. I'll just show you a couple. We did yeast nucleosomes. We did nucleosomes with a histone variant. We did nucleosomes with a methylation on this residue here. We did nucleosomes with a strange DNA sequence. And all of these nucleosomes look the same. And the RMSD of all of the ones that we solved is around 1.2 angstroms. So by all practical purposes, these structures are the same. So now to quote this famous artist, Gertrude Stein, I don't know what happened to my font here, a rose is a rose is a rose, loveliness extreme. Does the same hold true for nucleosomes? Or are we missing something? And so this got me to thinking about the limitations of X-ray diffraction. And so what's clear from our experiments is that nucleosomes that crystallize adopt the canonical structure. But what about nucleosomes that do not crystallize? How different are they? And how could we find out? And that's where we were lucky enough to engage in a collaboration with Jörg Langowski's group to use single molecule FRET to identify structural states of the nucleosome. And so the gist of this experiment is very criminally simplified here, that we put fluorophores, pretty much FRET pairs, pretty much anywhere on the nucleosome, on the DNA, on the histones. So we measure a lot of different pairs. We then measure the number of FRET events, particles that undergo FRET, and we do this at a number of different salt concentrations. And so this work is published, but the main conclusion from this is that we observe an open state that somebody, I think, called a butterfly state, which I really like. We observed an open state, and when you extrapolate to what we think are physiological conditions, about 1% of all nucleosomes at any given time are in this transient open state. And this is in addition to a state where the ends of the DNA partially unravel, as you can see on the left-hand side here. That's been identified many years ago by John Whittam and recently shown so beautiful by John van Noort's group and some other groups as well. And so this is an additional alternative state of the nucleosome. So this state exposes an H2A, H2B, an interface, a protein interface that's not open, not available in the closed structure. And so this has all kinds of important and interesting implications for histone variants, and we believe that the equilibrium, because these are in equilibrium, between these states is affected by epigenetic modifications, and this has very profound implications for DNA accessibility. Imagine if you're a polymerase and you're just cruising along and you're wanting to get access to that DNA. It's a very different matter whether you encounter the left state, the middle state, or the right-hand state. So, but to this audience, perhaps the more pertinent question is, does crystallography lie to us? Why don't we see this? And so the problem is that crystal formation favors the closed state. And that is pretty obvious when you look at a picture of the crystal packing of these structures, and you see this very close end-to-end packing. Base stacking interactions are very strong forces in crystallography. And so the crystal actually selects for this closed state. And so the important message here is that the very aspect that we're interested in, the dynamic behavior of nucleosomes, is in fact occluded by crystallization. So we had to find different techniques, and what is better than poor man's crystallography or small angle X-ray scattering? We call it poor man's crystallography because you do not require crystals, but you trade in resolution. So this is Bragg diffraction to the left. On the right is a Sachs pattern of a nucleosome. So you deal with very, very little information, but you can actually get out a radius of gyration of your particle, a maximum dimension, and if you do things properly and you trust your data, you can calculate an envelope that fits your molecule. And so this is what we did, and this is a test case of our best behaved nucleosome. And this pink envelope here is a de novo calculated envelope of the nucleosome without prior knowledge of the structure. And you see that the fit is actually really good. And so we really believe this proof of principle. So let's put this to the test. And so we applied this approach to one of our most difficult customers, the structure of a nucleosome with centromeric H3. This nucleosome had refused to crystallize, and now we know why. Because perhaps it is more dynamic than others, and so this would be a really good candidate. What is CEN-H3? Just a little bit of biology here. As our chromosomes divide accurately, the tubulin microtubules has to attach to the kinetochores. There's a kinetochore on each of the chromosomes, and then during the various phases of the cell cycle, pulls them apart. And every chromosome has this kinetochore, and the centromeric H3 variant, CENH3, is essential for kinetochore assembly. Now, the main reason why I'm showing you this picture is because I have serious microscopy envy, and I'm fortunate enough to have a brilliant colleague at CSU, Jennifer DeLuca, who works on this stuff, and so she gave me these pictures. All right, so the histone variant is essential for proper chromosome segregation. Why do we have a special variant at the centromere? How does it know to get there? And how does it affect nucleosomes? So let's address this last question here. And I should add here that the structure of these centromeric nucleosomes was extremely controversial. Some people said that it's a normal nucleosome, just maybe a tad different. Some people said it actually has lost its H to H to be dimers. It's just a tetrasome. Some people say it's a half nucleosome, just having one copy of each histone, and the DNA is organized in a right-handed manner. And some people say that H2A, H2B is replaced by another protein. So, of course, they can't all be right. And I think the debate is still open, and that's just for yeast, of course. So we decided to address this by what we do best, by in vitro analysis of reconstituted nucleosomes containing this histone variant. And so with data that I'm not going to show you, we showed that indeed, at least for our system in vitro, these nucleosomes form the canonical structure. They have all the histones in place, and the DNA is in the left-handed conformation. But when we applied Sachs to these structures, you can see that these nucleosomes are now quite bloated. Even if you don't believe the calculated envelope, which you can debate, the Dmax and the Rg is quite expanded, and this is very, very reproducible. So these nucleosomes are quite different. And through additional biochemistry, we've concluded that the ends of the DNA are actually quite a lot, are partially unraveled as shown in this picture to the left. But we actually did want more information on this, and so I was very lucky to talk Michelle Wang at Cornell University into collaborating with us, because in addition to microscopy envy, I also have severe single molecule envy. And so she developed this very elegant system where they tether DNA on their magnetic tweezers, and then they unzip the DNA double helix. So you unzip, 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 and then you encounter your nucleosome, and then you hit more resistance because the DNA has to unpeel from the surface of the histones before you can do further unzipping. So we applied this to our yeast nucleosomes, and very gratifyingly, she found that the terminal segments of the DNA are quite a bit weaker bound than the central turns of the DNA. And this, again, is just a half of a nucleosome. And we actually suspected this from just looking at the number of hydrogen bonds, but this was a very, very nice confirmation. And when you do the same experiment with nucleosomes with this histone variant that forms this bloated shape in Sachs, you can actually see that you need less forces than for the major type nucleosome to unravel these terminal ends of the DNA wraps. And so this was a very nice confirmation that these nucleosomes are in fact have ends of the DNA that are not very tightly bound. This seems to be a generally applicable finding because the Kuromizaka lab has recently published a structure of human centromeric nucleosomes where the ends of the DNA are completely disordered, and Tanya Pachenko and Ben Black's lab here at Philadelphia have had similar findings using a different approach. All right, so to summarize, this part is, and I'm really almost very close to being done, epigenetic modifications affect nucleosomes and their interactions. And so I want you to remember, whenever you see a nucleosome, forget those beautiful structures that are hanging around everywhere, but please keep in mind a more dynamic system, which is, I think, a much better reflection of what is really going on. These states are transient, and then they are in equilibrium, and we think they're influenced by post-translational modifications, by histone variants, and by DNA sequence. So rather than having a single structure, as would be suggested to us by our crystallography experiments, we probably have more of a range, a continuum of structures in various states of opening and closing. This is likely to have effects on interactions with other chromatin proteins and also will affect the way nucleosomes pack onto each other. So if you have nucleosomes that are very much in disarray because they kind of tend to be open a little bit more, you can imagine that these won't compact as enthusiastically as some others more stable, seemingly stable nucleosomes will. And this is something that we're actively pursuing. Okay, so the last brief segment deals with thermodynamics of nucleosome assembly and disassembly. And this is where I really learned to love fluorescence. It's very pretty, but it's also very versatile. And this work was initiated by Andy Andrews in my lab, and who's now at Fox Chase Cancer Center, and Aaron Heap, who's now in the job market. And so if you get his application, you should snatch him up. And Pamela Dyer did a lot of the work. Now, why should we care? This is a nucleosome model made by two very artistic students in the Bustamante lab. And you can imagine that the polymerase, oops, sorry, I thought this was a laser pointer, that the polymerase cruises towards this nucleosome and it's stopped in its track because it encounters this nucleosome where the DNA is bound to the histone octamer. And so it is very important for us to know what kind of resistance does this polymerase meet, and how is this affected by the various epigenetic modifications or by these various open states. But before we do this, we need to define nucleosome stability. How do we define nucleosome stability? Is it the propensity by which they open up and flop around? Or is it the propensity by which they shed their entire DNA and just make free DNA and free-floating histones? Or is it any of these states in between? Or any of these states here? So you can see from a thermodynamic viewpoint, there's a lot of work to do, and it's not simple work. But, so basically the point is here that nucleosomes are not binary systems, and there's many different ways to define nucleosome stability. Why is it so difficult to measure these rate constants? I mean, they're proteins and it's DNA, so what's the big deal, right? The problem is that histones are very basic, DNA is very acidic. Histones love DNA, and they don't greatly care whether they sit on it properly or not. And so we get all these non-productive assembly intermediates, non-canonical histone DNA interactions. Everybody who has worked with nucleosomes and histones in the test tube knows this. They are kind of messy to work with. And so at physiological conditions, if you just dump histones and DNA together, you get precipitate, because non-native histone DNA interactions predominate. How does a cell solve this problem? The cell actually has histone chaperones. This is a term coined by Ron Lasky in 1978. These are proteins that prevent the aggregation of folded histones with the DNA. So they're chaperones in the true term. And actually the term chaperones was first coined for these types of proteins and not so much for the folding chaperones. We have a lot of work with this histone chaperone. Let me see. This is supposed to spin. I don't think I can. Just a second. Yeah, okay. So this is a structure we solved a number of years ago, and this protein binds, called nucleosome assembly protein 1. It binds all histones with nanomolar affinity. It's found in complex with H2A, H2B in vivo, and it stabilizes a folded state of H2A, H2B, and this is unpublished work by Sheena Darcy in my lab. And it's been used for many years as a tool for nucleosome assembly. So how does it work? It actually allows the formation of the tetrasome, the first assembly intermediate. So this guy here, this one is supposed to form. And we can show this on a gel, and we can show this by the affinities. It seems that H3, H4 would much rather bind to DNA than to NAP, and that's a good thing. Okay. The situation is reversed, though, with H2A, H2B. If we just dump H2A, H2B onto DNA, these things are never going to make nucleosomes. And so NAP, they bind with about 20 nanomolar affinity, and under these same conditions, NAP1 will just pluck them off the DNA through competition. And this is what's seen in this simple gel assay here. We have histones bound to the DNA shown in this gel shift here, and then when you add the chaperone, it will pick them off. Okay, the same thing also holds true in vivo, and we've shown this in a very nice collaboration with my colleague Laurie Stargill at CSU, and this is published work. And so this seems to be a true function of this chaperone, to be kind of the garbage control and just cruise through the genome and pick up histones that are non-properly bound to DNA and to chromatin. But For us, this is a really useful tool, and Andy recognized that very early on. NAP1 mediates efficient nucleosome assembly through these propensities. We do this with fluorescently labeled histones, and you can see here, the more H2A, H2B we titrate in, the more nucleosomes we get, and you can prove through fluorescence and through FRET that indeed all the histones are present. And this situation looks very different when you do it under the exact same conditions in the absence of the chaperone. Everything works well till we hit about that magic mark of H2A, H2B. As soon as we add a little bit too much, boom, the whole thing goes into the gel well and precipitates out. And this is what NAP1 prevents. So we can actually do this assay also in solution to monitor the thermodynamics of nucleosome formation through FRET. So this is the system we're using. We can put our fluorophores on the histones, and then we get FRET as we form our nucleosomes as a readout, or we can put it on the ends of the DNA as an additional control. And again, as soon as a nucleosome closes in to form this nucleosome-like state, we can get FRET. So we can do this in 384 well plates. We can do this very pretty, it generates these very colorful scans. We can scan them on the Typhoon, get binding curves, and now we can actually go ahead and measure the effect of DNA sequence, and as shown in this example here, of histone modifications and a number of other things. And the beauty of this assay to me is that FRET always makes me a little nervous. You can change FRET due to a number of things, and not many of these reasons are good reasons. But we can take the contents of this well after scanning and run it on a native gel and convince ourselves that we really are making nucleosomes. And indeed, we could scan this gel, although with much higher error rates, and obtain KD binding constants from that. So we're now poised to do all these things, to measure the effect of all of these variations of nucleosomal component on nucleosome stability, but we're also very interested in the other side of the equation. How do histone chaperones work? Are all histone chaperones, do they function in the same manner? How do transcription elongation factors facilitate this dissolution of nucleosomes? What happens when a polymerase cruises through? And what happens after replication when chromatin is being assembled? So we now have the tools to look at all of these aspects. And we also are very interested in looking at higher order structure. Now, just to leave you with a big picture here, there's a number of ways to modulate chromatin structures. And I've shown you some that deal with variations of nucleosome components. We also work a lot on proteins that act on chromatin. And my point is, for example, histone chaperones, remodelers, nucleosome binding proteins. And my point is that we have to look at these phenomena in combination, because particular post-translational modifications may actually favor the binding of some remodelers and facilitate their action, or certain nucleosome binding proteins may inhibit or stabilize that open state, as we've seen. So there's a lot of work to do on the combination of this variety of modulators of chromatin structure. So I want to end by thanking the Luger Lab members, past and present, and I've highlighted people who did the work that I talked about. A lot of former members, all of them have successful careers in science, which is something that I'm very proud of, because I think that is really probably the most important thing that we do as teachers and professors. I also have wonderful collaborators, both at CSU and all over the world. And I talked about Jörg Langowski's collaboration and the collaboration with Michelle. So thank you guys if you're here. And then, of course, these wonderful funding agencies and the Biophysical Society that sustained me throughout all these years with their beautiful coffee cup, which is my favorite mug. And I have a little latte foamer in my lab, so thank you. And I just, sorry, I want to do a geography lesson, okay, because I get asked a lot. Anyway, so we live in northern Colorado, near the Wyoming border, not in Denver, not in Boulder. We live in Fort Collins, Colorado State University, Fort Collins, remembered. It's beautiful. It's been voted as the most livable city by Money Magazine a number of times. It's great. It's in the foothills of the Rocky Mountains, new building. We have great fishing, fly fishing, great kayaking, one of the best in the country. Rocky Mountain National Park, 45 minutes away from us. great natural prairie, untouched, never been farmed. Very, very interesting Native American history, very old sites there, and just a very nice place to live. So next time you're in the area, call me up and come and visit us and apply for jobs in our place. And with that, thank you all very much, and let's go party. I'd be happy to answer questions, if there are any.
Can I ask a question?
Yes.
You said about the centromere, and you asked a question, and maybe I misheard the answer. How did it get there?
Yes. So there are specific, so the centromere is a very, very complicated, sorry, the question was, how does the centromeric histone get to the centromere? The centromere is a very complex arrangement. The kinetochore, there's about 100 proteins that live there. There are specific assembly factors that specifically recognize the centromeric histone that deliver it there. And there's a number of labs, including ours, that work on this. In yeast, it's a little simple because in yeast, there's a specific DNA sequence that's very strange at the centromere. And that seems to be enough. In all other eukaryotes, no such thing. And so the centromeric histone epigenetically defines where the centromere is. So that didn't really answer your question, but that is because there is no answer yet. Other questions? Yeah. I can repeat.
So you convinced us the nucleosome is very dynamic. How much should we believe the structure of the nucleosome as relevant to chromatin?
Yes. Okay, so the question was that I've convinced you that the nucleosome structure is very dynamic, and should we believe the crystal structure of the nucleosome? Yes, absolutely.
Relative to chromatin?
Relative to chromatin. I think the majority of the nucleosomes are still in their nice, well-behaved shapes. Most of the time. We don't know time scales. We don't know how these things behave under physiological conditions. We don't know how factors affect that. And there's a number of people, very, very smart and brilliant people in this audience that address this using single molecule approaches. I would maintain that by and large, the structure is still true, but we have to entertain the notion that this is not a rigid structure, but that it can exist in this grayscale of dynamics. And that should not be surprising to us, because every protein that we know actually does this, as been shown by NMR work and HDX work and a lot of other people's work. So I'm not saying crystallography is lying. I'm just saying we have to consider it to be a little more mushy, if that's the correct word.
Can I have a question?
Where are you?
I'm here. I'm here. I can
yeah.
speak loud.
Okay.
Sorry. Yeah. So you show that the butterfly opening, but it happened at about, if I remember well, 500 or 600 millimolar sodium chloride condition.
Yes.
So have you ever seen that butterfly thing happen at physiology condition by some evidence?
Yes, so we can extrapolate back to physiological conditions. And so the numbers, the delta G that I mentioned, goes back to at physiological conditions, however you define it, we can say that about 1%, 0.3 to 2% of the nucleosomes are in these open conditions. You should probably talk to Jörg or Andy, who's somewhere in this audience.
Okay.
And they can give you the gory details of how they did this.
Yeah. Thank you for a wonderful talk.
Thank you. Sorry, I can't understand you. They're handing you a microphone. Sorry, I really can't hear you up here.
Okay. So nowadays there's more and more paper discussing about the red-handed nucleosome. This is
Yes.
the ribosome.
Yes.
So what do you think about the ribosome? And do you think there's evidence to prove the ribosome is really exists, and what's the biology function of ribosome?
So you're talking about the reversal of the DNA supercoil from
Yeah, this is-
left-handed to right-handed.
Yeah, and also your crystal structure shows that the nucleosome is left-handed.
Yes.
And why it cannot be right-handed? Why
Yes.
left-handed is more stable than the right-handed?
Yes.
And if there is a ribosome, what's the function? And yeah, thank you.
Yes, so it's true that all the crystal structures that we have are left-handed. It's also true that Ariel Prunell has shown many years ago that if you don't have the dimers present, that the tetramer can flip with a pretty low energy penalty from left-handed to right-handed. As soon as you get the dimers docked, it seems to me more frozen in place. Now, having said that, before we knew about these alternative open states, I would have said this is probably not going to happen with all the histones bound. But it's conceivable that when you have the butterfly structure, that you can get this flipping. I would probably think that the radius of gyration of these nucleosomes must be a little bit expanded. But I think there's increasing evidence for this reversal. As to what the biological implications are, I think there are many, because for a polymerase, you can imagine it would be much easier to have a reversed structure. And you would also expose different protein surfaces. So we could possibly, and we possibly have to add that structure to this whole zoo of alternative structures that I've already shown you.
Okay.
All right.
Let's thank Karolin one more time. That was
Thank you
great.
very much.